Hi everybody, my name is Alejandra Tapia Batres. This summer I had the honor of being in the Borner Lab at Indiana University. And today I'll be talking about the use cases for the Human Reference Atlas Organ Gallery. Oops, okay. So before I get into the use cases, I wanted to give a little bit of a brief overview about the various projects I was able to work on this summer. Unlike most of my fellow interns who probably had one major project or like one sort of research question that they were trying to answer, I was able to work on various projects. So in addition to the use cases, I also worked on an Oculus Store application for the Human Reference Atlas Organ Gallery. And going hand in hand with that, I was also able to create a documentation about methods we could use to track user engagements once that application is approved. I was able to also develop a hackathon website, and I also sat in and helped set up a usability study with our UX designer. I was also able to provide feedback for the CCF API in Jupyter Notebook, and I was also able to explore NewPrint and NewVid, which are tools that we could hopefully use in the future for inter and intracellular interactions. And so this diverse set of projects during my HuBMAP internship has contributed to various aspects of the HRA Organ Gallery. And this basically just demonstrates the multi-disciplinary nature of HuBMAP and its efforts to make valuable biological data accessible and applicable to various scientific fields. All right, so as I mentioned earlier, I'll be talking about the Human Reference Atlas Organ Gallery in VR. But before I do that, I want to give a little bit of a background about how we get those organs into VR. So first, organs start off at tissue centers where they're collected from donor bodies. And then assays and analysis are performed to ensure the tissue is healthy. And this data is compiled at multimodal imaging centers. So we can see here in picture A to the left-hand side, we have a photo of a reference organ. It's the human male left kidney on a cutting board with spatial markers. And commonly the 3D size, position, and rotation of a tissue block is captured by taking these images of the tissue dissection process. But it's also difficult to compute the exact spatial location, rotation, or size of extracted tissue blocks from these photos. And then in photos B and C, we have the registration user interface, better known as RUI, which was designed for usage by experts who collect these tissues and need to document the tissue extraction site. And so I'll be talking about RUI in a little bit later on in the presentation. So in order to integrate data from the various bodies, a common coordinate framework is created. This is the CCF. So the tissue blocks need to be registered and mapped, so to speak, to get to the organ system framework. And this basically defines spatial positions of major anatomical structures and cell types within the 3D reference body. And so this is super valuable because if other tissues are available from organs that are not in the CCF, they can later on be added on. All right, so here we have a screen recording of RUI, the registration user interface. And so that yellow block there represents a tissue block. So that way our experts are able to go into the reference organ and place where that tissue block sample goes. And so the Human Reference Atlas is basically a comprehensive 3D atlas of all the cells in the healthy human body. And this was made possible by the collaboration of multiple experts who have developed standard terminologies that are linked to these 3D reference objects. And so I have a Nature paper listed here in case anybody's interested in learning more. And then the Exploration User Interface, also known as the EUI, allows exploring the Human Reference Atlas to be done spatially and semantically. And so here we have a screen recording. It's starting at the end for some reason. So I'll give it a sec to go back to the beginning. So you can kind of see what happens when you go into the EUI portal. So here we have the human and the male and female human body. And here I'm zooming into a specific organ. And once you're zoomed in, you're able to see the various tissue blocks that are registered. And so once you click on a block to the right-hand side, you can see that it actually takes you directly to the HuBMAP portal, where you're able to see not only the sample ID, but additional metadata of that tissue block. And so I have the website listed down below in case anybody's interested. And so basically, if we take the RUI and the EUI from a 2D screen and convert it to virtual reality, we have the Human Reference Atlas Organ Gallery. And so here we have a screen recording of myself in virtual reality. You can see the various organs there with their titles. So it's very trippy when you first get into it. And in just a moment, I'm going to turn around and you can see there the male and the female human body. All right. And then here you can see my red pointer there. I'm clicking on tissue blocks and you can see to the left-hand side, there's a little bit of like a little white card. This shows static data, which includes not only the HuBMAP ID, but also if it's not from HuBMAP, because this is a collaborative effort. So we have received tissue samples or tissue blocks from other collaborators, and it also lists the predicted cell types at the bottom. So with the use cases for the organ gallery, these are basically user stories compiled out of interviews with 17 experts across the HuBMAP consortium. And so there were two main goals with this use case study. The first was to obtain guided input regarding existing use cases for the organ gallery. And the second goal was to generate new use cases through the input provided by the interviewees. And so we already had 3 existing use cases, which were the quality assurance / quality control, onboarding to the HRA, education and outreach. And then through the interviews, we were able to identify 5 additional use cases. We have embodied data stories / patients, which would basically utilize VR to enhance patient education, especially when it comes in relation to their anatomy and functionality after undergoing medical procedures. We have animations, where we would utilize animation to enhance the understanding of the body's mechanisms and disease processes. We have histology, where experts would be able to analyze tissue and cell profiles. We have multiplayer, where people would be able to interact with with multiple people in the organ gallery. And then we have spatial RNA data, where experts would be able to explore and analyze cellular landscapes. And so out of these 8 use cases, in regards to the organ gallery, we'll be focusing on onboarding to the HRA and the embodied data stories. Oops. Okay. And then here we have a screenshot. As I mentioned earlier, one of the things I worked on was an application for the Oculus Store to have the organ gallery be available to the public. So we can see here, it looks very different from the screen recordings I did earlier. The background is different. The human bodies are white instead of blue just because of aesthetics. So one of the next steps is to make those bodies transparent so the organs are more visible. And going hand in hand with that, the methods for tracking user engagement once the application is approved include user analytics, where we'll be able to collect demographics and usage patterns of people that download the app. We have time-based metrics, where we would see how long people are spending on the app or how long they take to perform specific tasks. We have performance metrics, where we would see the frame rate or loading times. We have event logging, where we would be able to see what a user is doing, if they're just smashing buttons, or what menu selections they're doing. We also have user survey / in-app feedback. You know, like after you download a new app, it usually pops up like, do you love our app? Leave us a review, that kind of thing. And then we have heat maps, where we would be able to understand how users interact with the app. And last but not least, we have good old social media. You know, if you're enjoying the app, you want to share it with your friends. And so when I first came into this internship, I had absolutely no computational experience. And now I'm leaving with not only experience with like things such as Unity, GitHub, Blender, Visual Studio Code, and learning programming languages like HTML, Python, C# and R. But I'm leaving with a more confident self than I came in. I'm starting my master's program in about 3 weeks for bioinformatics. And so I'm really excited actually to do that. And so this internship has not only helped me prepare for grad school, but it's going to help me with my career goal of becoming a bioinformatician. And so lastly, I would like to thank everybody in the Red Pill, Blue Pill VR R&D team for all their efforts thus far. Thank you to my mentor, Andreas Bugle. He made this internship such a great time. Thank you to my PI, Caddy Borner, and shout out to CNS and Indiana University for hosting me this summer. It was a great time. Thank you to HuBMAP for giving me the opportunity to have this internship, and thank you to NIH for funding HuBMAP. And then we have some relevant papers in case anybody's interested. But yeah, thank you guys all so much, and I'm happy to take any questions.
Wonderful presentation, and we have